White and rosé winemaking requires particular attention to the risks associated with oxidation phenomena. Color and aroma are key determinants of wine quality and freshness for these wines, and oxidative stability is therefore at the heart of winemakers' concerns, especially given the current trend to limit chemical inputs, particularly sulfites.
Contact between oxygen and wine is impossible to avoid, not only during winemaking, but also after bottling. Small amount of oxygen goes through the closure inside the bottle. Oxygen doesn't react by itself, but the presence of transition metal, such as iron or copper, promotes the production of radical oxygen species or quinone. Quinones are oxidized form of catechol, which is present in most of the polyphenolic compounds. These quinones are then involved in further reaction, causing slow modification of color and aromas in wine.
Glutathione is a natural antioxidant present in grapes and in yeast namely, that acts as a quinone scavenger. In this context, Lallemand was the first at developing and proposing inactivated yeasts rich in glutathione for winemakers about 15 years ago. Recently, a considerable improvement in the levels of reduced glutathione has been achieved through the use of a yeast strain selected for its unique properties and leading to the development of the inactivated yeast GLUTASTAR. In numerous trials run at lab, pilot and industrial scale, when added at the earliest stages of winemaking at the press outlet, GLUTASTAR proved to be efficient at protecting the aromas and color of wines up to the latest stages after bottling. Suspecting that the efficiency of glutathione-rich inactivated yeast was not only relying on their glutathione content, and with the objective to better understand the action mode, Lallemand started a research collaboration 5 years ago with Régis Goujon's team at EUVV in Dijon.
At EUVV, we've been particularly concerned with premature oxidation of white wines for more than 15 years now. To that purpose, we've developed an innovative analytical approach to understand the chemical mechanisms involved throughout winemaking, and in particular to understand how wines can resist to oxidation. This approach is called metabolomics. It has been developed in very close collaboration with Professor Philipp Schmidt-Koechlin at Technical University of Munich.
The mass spectrometry measures the mass of ionizable molecules in a solution. Thanks to the mass and the isotopic profile, it could be possible to annotate ions with their elemental formula. The pool of ions in a solution produces an instant chemical picture of the samples. The metabolomic analysis reveals the diversity of compounds released by inactivated yeast. These compounds can be plotted depending on their molecular formula. GLUTASTAR, which combines a unique yeast strain and an optimized production process, exhibits a higher diversity of sulfur-containing compounds. These compounds can be involved as nucleophilic competitors and thus protect wine color and aromas.
Finally, implementation at the earliest stages of the white or rosé vinification process ensures better preservation of color and aromas throughout the process and up to bottling. Beyond the high content of reduced glutathione, the wealth of reducing compounds explain the anti-radical activity of this tool. Thus, the combination of glutathione, a trap for quinones, with other active compounds that are traps for free radicals, makes it an innovative tool of choice for the control of wine aging.